Hello everyone! Today we are going to talk about glycogen metabolism, both synthesis and breakdown. So here we are in our study of carbohydrate metabolism. We've already talked about gluconeogenesis and we will talk about the pentose phosphate pathway later. So today we want to talk about glycogen metabolism. How do we synthesize it and how we degrade it? So I wanted to show you this graph right here because this shows what happens if an individual undergoes prolonged starvation. So this red curve right here shows the y-axis shows the glucose used in grams per hour. So here you have absorbed glucose from your food, so it's pretty high. But if you don't eat again, then what happens is glycogen is then broken down. And then that lasts for about a little bit more than maybe 24 hours. And then gluconeogenesis ramps up. So if the starvation is prolonged, then you would break down fat. But we'll talk about that in chapter 17. So where are we? We are here. So if you are a plant, you make starch, but if you are an animal, you make glycogen, so our focus will be on glycogen. So glycogen is stored in the liver, and the reason that glycogen is stored there is that the job of the liver is to act as a glucose buffer. So if we are eating a lot, the job of the liver is to take up glucose and synthesize glycogen. But if we're not eating, then the liver's job is to break down glycogen and then provide glucose to the blood, and then other body tissues will take up that glucose. And that's important because the brain and red blood cells rely entirely on glucose. So we want to talk about glycogen breakdown in the liver. We also have glycogen in the muscle, but here the role of the glycogen is different. Structurally, it's exactly the same, but the role is different. The glycogen in muscle is degraded to provide energy to the working muscle. So what happens is glycogen is degraded and it doesn't go all the way to glucose, it just goes to glucose 6-phosphate. And then glucose 6-phosphate does not leave the muscle cell. It remains in the muscle to enter glycolysis to provide ATP for the working muscle. So in the case of the liver, the final breakdown product is glucose, but in the case of the muscle, the final breakdown product is glucose 6-phosphate. So let's review our glycogen structure. Remember, it's a polymer of alpha D glucose. We have our alpha 1,4 glycosidic bonds here and our alpha 1,6 glycosidic bonds here. So we have our branch points. And again, glycogen is stored in the muscle and in the liver. Remember when we talked about glycogen structure, we talked about reducing and non-reducing ends. So our non-reducing ends are here. We only have one reducing end. And in fact, if we have a very large, that's our non-reducing ends, if we have a very large polymer, you can see that you only have one reducing end, but you have lots and lots and lots of non-reducing ends. So that's important because there's interesting chemistry will happen at the non-reducing ends. So having a branched polymer gives you lots of non-reducing ends and allows those reactions to occur at the non-reducing ends, such as glycogenolysis. So glycogenolysis is just lysis of glycogen or breaking down of glycogen, and we're going to do what's called a phosphorylasis. So we've talked in the past about hydrolysis, and hydrolysis uses water to hydrolyze bonds. Here we're going to use phosphate to break bonds. So instead of doing a hydrolysis right here, we're going to do a phosphorylasis. So if we use inorganic phosphate and we do a phosphorylasis, then what happens is we have glucose 1-phosphate as our product. And our glycogen now is one fewer residue than it was before. So this happens at the non-reducing end. The enzyme that catalyzes this reaction is called glycogen phosphorylase A. So A stands for active. And we have both the delta G0 primed and the delta G values right here. So you can see that the delta G0 primed is very slightly positive, but that's standard conditions which don't exist in the cell. In the cell, the delta G is a -5 to -8 kilojoules per mole, so that means it's a favorable reaction. So the advantage of phosphorylasis over hydrolysis is if you broke this bond with water, you would just end up with just plain glucose, and then you'd have to spend an ATP to phosphorylate it. But here, we don't have to. We already have our glucose 1-phosphate is already phosphorylated. So this saves us an ATP. So if we look more at a big picture, think of glycogen with lots of its branch points and lots of non-reducing ends. And if I'm going to do a little animation right here where I just show we can have, say in this picture, we'd have 2 copies of glycogen phosphorylase A, and each one of them is sort of nibbling away at the non-reducing ends until it gets to a branch point. So this part is pretty fast. But what happens is when you get to within 4 glucoses of a branch point, the reaction stops. This is a picture of glycogen phosphorylase A right here. And it's a dimer, so its catalytic site is right here. It also has an allosteric site, which controls its activity. But what happens is you're feeding the glycogen into this really narrow cleft right here. And as soon as you have a branch point that bumps up into that cleft, then it won't work anymore. So you get to this point and then you stop. So what happens next? Well, luckily we have another tandem enzyme. So this is a tandem enzyme, so it has 2 activities on the same polypeptide chain, and we have a transferase activity that takes 3 of these glucoses and moves them over here. So now we have those 3 glucoses right here. And then we have a glucosidase activity that just does a hydrolysis and pops off that glucose. So now our branch point is gone. So the glycogen phosphorylase A can come back and it can do the phosphorylasis one by one, and it will stop when it gets to the next branch point, and then we will call upon these 2 enzymes to get rid of the branch point. So it is all right with me if you want to combine the names of these 2 things and just call it a debranching enzyme. So it's fine for us to call it that. Okay, so when the glycogen phosphorylase A has done the phosphorylasis and then the branching enzyme has helped out by getting rid of the branch points, we have a big pile of glucose 1-phosphate. And remember, this mutase reaction is reversible. So all of the glucose 1-phosphate is isomerized to glucose 6-phosphate. So we'll go in this direction. It's a reversible reaction. It will go towards the left if you have lots and lots of glucose 1-phosphate. So the glucose 1-phosphate is converted to glucose 6-phosphate. The enzyme is a mutase. Remember, mutase is a special type of isomerase where you move a phosphate from one position to the other. So here we move the phosphate from the number 1 position to the number 6 position. And then glucose 6-phosphate is linked to all of those other pathways that we've talked about before. I did want to show you this. There's a picture of a stamp that came out in 2008 with Gertie Corey on it. So they celebrated her work with carbohydrates with a stamp. Okay, so now what happens? If you're in the liver, you have glucose 6-phosphatase. So you might remember seeing glucose 6-phosphatase before. This is an enzyme that's part of gluconeogenesis. And the liver carries out gluconeogenesis as well. So this is a hydrolysis. We remove the phosphate and now we have plain glucose. So this is necessary because the phosphate's negatively charged, so we have to take the phosphate off so that we can get the glucose to go through its transporters. The transporters wouldn't recognize glucose 6-phosphate. And glucose 6-phosphate being negatively charged is not going to pass them through the membrane. So now we have plain regular glucose, and now the glucose leaves the liver and enters the bloodstream, and it's delivered to other tissues. So the same process happens in the muscle, except for the last part. So both the liver and the muscle have glycogen phosphorylase A, and again, A stands for active. They both have this debranching enzyme that can get rid of the branches when the glycogen phosphorylase bumps up into a branch point. They have this phosphoglucomutase that can convert the glucose 1-phosphate to glucose 6-phosphate. The liver has the glucose 6-phosphatase. The muscle does not. The muscle doesn't express it. So with the glucose 6-phosphatase in the liver, the glucose 6-phosphate is converted to glucose, and that means the glucose can leave the liver, enter the blood, and other body tissues can take up that glucose. But the muscle hogs the glucose for itself. In fact, it doesn't get to glucose at all. It just gets to glucose 6-phosphate. And then glucose 6-phosphate just hops into glycolysis and then is oxidized to provide ATP to power the working muscle. So no glucose 6-phosphatase in the muscle. So of course we want to talk about how these pathways are regulated, and we will do that in a minute. But I want to focus your attention on the left side. So on the left side of this figure, um, this is a summary of glycogen breakdown. So glycogen was broken down by a phosphorylasis using the enzyme glycogen phosphorylase A and with the debranching enzyme to help out the glycogen phosphorylase. You get a whole pile of glucose 1-phosphate and the mutase converts it to glucose 6-phosphate. And we said earlier that the delta G is -5 to -8 kilojoules per mole. Well, now we want to talk about glycogen synthesis. How are we going to take glucose 6-phosphate and convert it to glycogen? Well, we can't reverse the reaction because it would be positive. So it must occur by a different pathway, and it does. So let's talk about that. But let's first do our where are we? So glycogen synthesis, these are our anabolic pathways. So here we want to, when we're fed, to take glucose and synthesize glycogen in glycogen synthesis. So again, if you were a plant, you would take glucose and you would make starch. So how are we going to do this? Well, we can't just take glucose 6-phosphate and then just stick it on the end of a glycogen. That's not going to give us a negative delta G. So we are going to use the mutase. So here we have glucose 6-phosphate, and remember that that reaction was reversible. So we're going to make glucose 1-phosphate. So we have glucose 6-phosphate shown here makes the glucose 1-phosphate that's shown in this figure in purple. Then the next thing that we're going to do is we need to synthesize activated glucose. So we're going to use UTP, energetically equivalent to ATP, and this phosphate this oxygen, is going to nucleophilically attack this phosphate, and then we are going to have pyrophosphate as a leaving group. And what that means is that we have UDP glucose. So here's our glucose 1-phosphate. So this purple phosphate was the phosphate that was already on the glucose 1-phosphate, and then this phosphate right here is on the UTP, and then here's the ribose and the uracil. So we end up hydrolyzing 2 phosphoanhydrate bonds, one here and one here. But since we form one here, we would really say the net is the hydrolysis of 1 phosphoanhydrate bond was needed to convert glucose 1-phosphate to UDP glucose. So when you synthesize glycogen, every single glucose 1-phosphate that you start out with has to be converted to UDP glucose, and every single one of them will cost you the hydrolysis of 1 phosphoanhydrate bond. So again, we hydrolyze this phosphoanhydrate bond here and that phosphoanhydrate bond here, but since we also form one, the net is 1 phosphoanhydrate bond hydrolyzed. And there it is in green. So if we look at the energetics of this reaction, the glucose 1-phosphate and the UTP give us our UDP glucose, and then pyrophosphate. And then the pyrophosphate is hydrolyzed to form regular phosphate. And again, we form this delta G is 0 because we've hydrolyzed a phosphoanhydrate bond, but then we formed a new one right here. So we need to hydrolyze an extra one in order to get this reaction to be spontaneous. So here I have the delta G 0 primed and not the delta G, but the delta G is also a high negative number. So the first step is the UDP glucose pyrophosphorylase enzyme, and the second step is pyrophosphorylase. And again, one net phosphoanhydrate bond, which is shown right here, must be hydrolyzed in order for this reaction to be exergonic. Okay, so now we have our UDP glucose. So that's shown right here. So it just shows this hexagon is the glucose, and there's the UDP. And what happens is we're going to add this glucose onto a pre-existing glycogen at the non-reducing end. So again, that's nice. We have this branch glycogen with all these non-reducing ends. So what happens is we're going to tack this purple glucose at the end of this glycogen, and then UDP is going to be the leaving group. So again, we're going to um, do another hydrolysis of a phosphoanhydride bond. And the enzyme here is called glycogen synthase. So we need pre-existing glycogen, we need UDP glucose. Regular glucose would not work. We couldn't just tack another glucose here. It would have a positive delta G and that wouldn't work. So this shows the chemistry right here. So here's our UDP glucose right here, and then this shows the hydrolysis right here, and then UDP is our leaving group. So we form this oxonium ion intermediate, and then here is our pre-existing glycogen. So the OH right here, that's carbon number 4, nucleophilically attacks over here, and that's our oxonium ion intermediate, and now we have formed a brand new alpha 1,4 glycosidic bond. So here is our glycogen. It is now longer by 1. We've added a glucose on it. So that's what we do. We just take a whole lot of UDP glucose and we just keep tacking on the glucoses onto the non-reducing end. So we did say that we need pre-existing glycogen. So how do you get started if you need pre-existing glycogen? Well, this is how it works. This is how you prime it. So this little circle right here is glycogenin. This is what it really looks like. So glycogenin is a homodimer, and it's shown here in each of the monomers. There shows a UDP glucose. So there's an active site there, and there's an active site there. So what happens is there's a tyrosine that is shown. It's kind of hard to see, but there's a tyrosine on the protein, and tyrosine, remember, has an OH group. So what you can do is you can start building your glycogen on the tyrosine. So the glycogenin then it's an enzyme, but it's also a scaffold. So what you do is you start here with the tyrosine and then you add 7 glucoses. And then once you have gotten to your 7 glucoses, then glycogen synthase can take over now. That gives you enough of your pre-existing glycogen. So then here's your glycogen right here. So now the glycogen synthase is able to add all of these glucoses to form these long chains. We're going to need a branching enzyme though to help us out here. So this is how the branching enzyme works. So basically, if we're adding glucoses one by one to the non-reducing end, pretty soon our glycogen is going to get pretty long. So what happens is the branching enzyme, what it does is it will take it will just sort of clip off some of these glucoses and then add a branch right here. So this shows a glucose with one branch, and it shows us creating a second branch. So this one got kind of too long, so we're going to clip off a whole bunch of glucoses right here, this chain, and then we're going to synthesize an alpha 1,6 glycosidic bond right here. So now we have 1, 2, 3 non-reducing ends, so we can have 3 copies of glycogen synthase adding on the glucoses to make it longer and longer. And as soon as they get a little longer, then the branching enzyme will come and snip some of that off and add another branch. So at this point, since you're watching this, you will have done your top hat quiz. So what we want to do is we want to talk about how these pathways were controlled. So the question I asked you is, what enzymes do you think would be controlled in degradation and in synthesis? So here are our answers. We're going to control the glycogen phosphorylase in the degradation process and the glycogen synthase in the synthetic process. So all of these other enzymes are important and needed and all of that, but they're not going to be controlled. So how do we control them? Okay, so we need to put this into a physiological context. So when energy is required, remember, think of the 2 places where we have glycogen. We have it in the liver and we have it in the muscle. So we want to break down glycogen when we're fasting because the liver will give the blood glucose when it's needed. We also need to break down glycogen when we're exercising. So in both of these cases, we're going to want glycogen phosphorylase to be active. On the other hand, if we're resting or fed, it will be the opposite. When energy is available, such as when we're fed, then we can take that glucose from our diet and we can use that to synthesize glycogen in the liver for the hard times to come ahead. And the same thing for the muscle. If we're resting, then we can use glucose from our diet to synthesize glycogen, and then we can call upon that the next time we do some exercise. So here, the main way that we control these enzymes is by phosphorylation. So phosphorylation is under hormonal control. We're also under allosteric control. Allosteric control is sort of a fine tuning. So phosphorylation allows the enzymes to be more sensitive to allosteric modulators. So we're going to use the form A for active and B for inactive. What makes this a bit confusing is sometimes phosphorylation will make an enzyme active, and sometimes phosphorylation makes it inactive. It just depends on the enzyme. So let's see if we can make sense of this. Okay, liver, fasted state. Okay, so the hormones that are secreted in the fasted state are both glucagon and epinephrine. So glucagon from the pancreas and epinephrine from the adrenal glands. So they are, again, our glucose sensors, and they notice when you're fasting and your blood glucose concentration goes down. So they both act on the liver in the same way. So glucagon binds to a glucagon receptor, and epinephrine binds to an adrenergic receptor. So adrenergic comes from adrenaline. You might know that epinephrine and adrenaline are the same, that they're the two names for the same thing. So we talked about how glucagon turned on gluconeogenesis. Glycogenolysis is going to help us out because both gluconeogenesis is going to make us glucose from non-carbohydrate precursors, and glycogenolysis is going to break down glycogen to glucose. And in both cases, we get glucose, and the glucose can go through its transporter. So this is a GLUT2 glucose transporter, and the glucose can leave. So in the fasted state, glucagon and epinephrine are released from the glucagon from the pancreas and epinephrine from the adrenal gland. So what we do is glycogen phosphorylase needs to be on in the fasted state, and glycogen synthase needs to be off. If we're fasted, this is no time to be making glycogen. We don't have the resources to do that. So a question I asked here, and hopefully you got the answer, was the answer is both. Both of these enzymes are phosphorylated. You might remember the last time that we talked about gluconeogenesis is we talked about how glucagon binds to its receptors, activates the G protein, activates adenylate cyclase, produces cyclic AMP, and cyclic AMP activates protein kinase A. And protein kinase A does what kinases do, and it phosphorylates targets. So both the glycogen phosphorylase and the glycogen synthase are both phosphorylated. But in the case of the glycogen phosphorylase, phosphorylation turns it on, whereas in the case of glycogen synthase, phosphorylation turns it off. So what we have to remember is in the fasted state, we have cyclic AMP. And cyclic AMP activates protein kinase A. And protein kinase A phosphorylates things. So we're going to see a lot of phosphorylated enzymes. And then we should think, what enzymes am I going to want to have on in the fasted state? And what enzymes am I going to want to have off? So it makes sense for glycogen phosphorylase to be on and glycogen synthase to be off. So here's another picture from another book. So here's glucagon. It binds to its receptor, activates the G protein, which isn't shown here, activates adenylyl cyclase, and then ATP produces cyclic AMP. Cyclic AMP binds to protein kinase A, pulls away those regulatory subunits, and now we have active protein kinase A. So now there's another little wrinkle right here. Protein kinase A actually doesn't phosphorylate glycogen phosphorylase directly. What happens is the protein kinase A phosphorylates phosphorylase kinase, which is another kinase, and then phosphorylation turns it on, and then phosphorylase kinase phosphorylates glycogen phosphorylase, and phosphorylation turns it on. And when All these names sound the same. When glycogen phosphorylase is active, then what will happen is glycogen will be broken down and you will get lots and lots of glucose 1 phosphate that can be converted to glucose 6 phosphate. And if you're in the liver, which we would be because that's glucagon, glucose 6 phosphate is converted to glucose. It leaves the liver to go to other body tissues. So again, fasting, glucagon binding to its receptor results in the activation of protein kinase A. And in the end, the activation of glycogen phosphorylase A occurs as a result of its being phosphorylated. So here's glycogen phosphorylase. So it is a dimer, and so we have a T form and an R form. So here's the T form right here, and there are serine residues that are that can be phosphorylated. So in the T form, the serine's not phosphorylated. But when the serine is phosphorylated, look at there's a huge conformational change. The serine, there's a huge shift where the serine right here the serine the serine 14 was way down here, but once it becomes phosphorylated, it's way up here, and that causes a conformational change that converts the enzyme from the T form to the R form. So there's the active site right there. In this T form, the conformation is such that the enzyme can't work, but when you've got this huge conformational change that occurs once the enzyme is phosphorylated, now the active site can bind substrate and the reaction can occur. So what else is happening at the same time? Okay, we're still fasting. We want to make sure not only that the glycogen phosphorylase is on, but that the glycogen synthase is off. So because the glycogen synthase would be synthesizing glycogen. We can't do that when we're fasting. So again, when glucagon binds to its receptor and activates the adenylyl cyclase and then produces cyclic AMP, so protein kinase A is active, one of the other targets is glycogen synthase. And here, glycogen synthase is inactive when it's phosphorylated. So phosphorylation turns glycogen synthase off. So we have glycogen synthase B. Remember, we're using A for active and B for inactive. So we're not making new glycogen. Glycogen synthase is inactive. When you're fasting, you're not able to make new glycogen. So we're fasting, or maybe we're exercising. So glucagon doesn't act on the muscle during fasting, but when you're exercising, the epinephrine does. So epinephrine is secreted by the adrenal glands, and it binds to adrenergic receptors in the muscle. And here, glycogen is broken down to glucose 1 phosphate and then glucose 6 phosphate. That's as far as you get. So the glucose 6 phosphate then hops into glycolysis to make ATP to power the muscle. Notice that nobody is leaving here. All of the glucose 6 phosphate that results from glycogen breakdown in the muscle stays in the muscle. Okay, so you fasted or exercised or both, and now it's time to eat. Okay, we're going to rest and we're going to eat. So what's going to happen now? Well, when we're eating, those glucose sensors will notice that we're eating and our blood sugar is going back up because now we have glucose from our diet. So glucagon will no longer be made and no longer be secreted from the pancreas. Instead, we're going to be secreting insulin. So now we have to turn off all of those signals we just turned on. And one of the things that we need to do is that cyclic AMP, we need to get rid of that. So there's a phosphodiesterase enzyme that will just catalyze a hydrolysis. And then we convert cyclic AMP to just regular old AMP, and AMP is not a second messenger. So you might remember from earlier, the other thing that we talked about was that insulin activates phosphoprotein phosphatase 1, and the phosphatase enzyme removes all the phosphates. So all of those phosphates, the protein kinase A put on, the phosphoprotein phosphatase 1, the PP1, will take them right off. Okay, so now we're fed. So insulin stimulates glycogen synthase. So in the pancreas, we will secrete insulin and insulin will bind to its receptor. And we won't talk about the details here because that's beyond the scope of this course. So I wanted to remind you what enzymes and what processes are active in the fed state. So in the fed state, we want synthesis to be active. So we want glycogen synthase to be active. We don't want glycogen breakdown to be active, so we want the glycogen phosphorylase to be inactive. So here's the glucose from our diet. It comes into the liver through those GLUT2 transporters, and we're going to use that to make glycogen. In the muscle, we have GLUT4 transporters, and insulin stimulates the appearance of those transporters on the muscle cell. So the insulin's job is to make sure that the glucose transporters that would be on the inside of the cell are transported to the plasma membrane so they can bring glucose in. So insulin is absolutely necessary for that to happen. So we will take our dietary glucose and we'll use it to make glycogen for the muscle now that we're resting, for the next time we need it for when we're exercising. Okay, we're in the fed state now. So we're in the fed state. We want the glycogen phosphorylase to be off and the glycogen synthase to be on. So we have our hormone now is insulin. So the glycogen synthase is active. So I've colored it in green here. Glycogen synthase is on, glycogen phosphorylase is inactive, and glycogenolysis is off. So now my question was, what enzymes are dephosphorylated? Well, it's both of them. Okay, so we're going to take the phosphate group off of both of these enzymes. So the protein phosphatase 1 that was activated by insulin will remove the phosphate groups. So now glycogen phosphorylase was active when there was a phosphate on it, but now that you take the phosphate off, it's not active anymore. And remember, glycogen synthase was active glycogen synthase was inactive when it was phosphorylated, but now that we take the phosphate off, the glycogen synthase is on. So I have a little summary, a little animation right here. So let's go down here. Okay, so here we are in the liver cell. And if it's glucagon, we must be fasted. So glucagon binds to its receptor, activates the G protein, activates adenylate cyclase. Adenylate cyclase catalyzes the synthesis of cyclic AMP. Cyclic AMP activates protein kinase A and puts together that cascade we talked about. Glycogen is degraded all the way to glucose, and glucose leaves. So epinephrine can do that too, and we're not going to talk about the details either, but there are actually two kinds of adrenergic receptors. There are alpha and beta adrenergic receptors. The beta adrenergic receptor acts the same way as glucagon. It results in the activation of cyclic AMP. The alpha adrenergic receptor results in the release of calcium into the cytosol. Anyway, both of these things collaborate again into activating glycogen phosphorylase so that glycogen can be degraded all the way to glucose and glucose leaves. Okay, so if we are fed, then glucose will come into the cell when we are fed. This will be from our diet, and then glycogen synthase will take place. And then this will actually inhibit the phosphorylase to stabilize the T form, because if we're fed, we don't want the glycogen phosphorylase to be active. Okay, now we're in the muscle cell. Remember, things are a little bit different here. So we have the beta adrenergic receptors. They act just like glucagon. They result in the increase in cyclic AMP. And glycogen is degraded to glucose 1-phosphate and then glucose 6-phosphate. And then glycolysis uses that glucose 6-phosphate to make ATP to the muscle. So notice that nobody is leaving. When we are resting and fed, then the insulin receptor will activate. This is GLUT4. It's not labeled that way here, but that's what it is. The glucose again will come into the cell when we are fed, and then glucose can be used to synthesize glycogen. All right, let's go back to our PowerPoint. So much the same thing is happening here, although I like this figure because it shows what's happening to insulin. So what happens when we are fed is that insulin activates this protein phosphatase 1, and what's being shown right here is that the protein phosphatase 1 is taking all of the phosphates off by hydrolysis. So it takes the glycogen phosphorylase A, takes its phosphate off. Now glycogen phosphorylase is glycogen phosphorylase B. It's inactive. It inactivates the phosphorylase kinase by dephosphorylating it, but it activates the glycogen synthase by taking off its phosphate. And now you can synthesize glycogen. Well, I hope you enjoyed this whirlwind tour through glycogen synthesis and degradation, and we will talk again next time.